church that we knew. Well, I said, well, you know.
Hey, Ella, doing a sound check. Can you hear me?
Wrong, now I have to go home with.
Yes, I can.
All right, great. Thanks very much.
I'm so excited about this program. It sounds so interesting. Thanks for zooming it.
Yeah, no problem.
Stay overnight Saturday night.
Oh, good. We got our speaker on.
Yeah, I think it's supposed to take rough for me.
Friday.
And I think it's working night.
Yeah. So, yeah.
Hey, Dr. Nigger, I'm Nick.
Mini farm.
Yeah, so did I.
Hi, can you hear me?
I can, yeah. Thanks for participating in the early sound check.
Okay. Yep, I think I'm all ready to go here. Might as
Alright.
well see.
I'm gonna make you a co-host.
Yep. I think we're good.
That's how I hope it is.
Or, let's see here.
I don't want to just.
Oh, hello. Yep. I want my hands and how it's going. Yep.
I am opposed.
I got it.
Oh, you gotta do it? Oh, okay, cool. All right, so someone's making you a co-host. I can't right this second, but Tom's about to. So, yeah, we'll have you set up shortly, and I think Tom will let you know when you're on, but I think like 15 minutes in was discussed or something like that.
Yep, sounds good.
Okay, great. Yeah, feel free to, mute the video off or whatever until you hear one of us call.
Sounds good.
Thank you.
My knees were out. I thought they'd be out because it was nice and warm. I think what it is. It was more warm than, what, 2 weeks ago? They all got back there.
I need your Danny back, huh?
That's what I'll find them, get one, I only want to do, but you only need one, yeah, probably. I was still.
What tree are you taking down, Tom?
They
Have two soft maples.
The bees, because.
Two big socks. I don't know.
Okay, yeah.
I'll get it going for you there.
That's right. Yeah. He went to it now.
Two giant soft maples.
Okay, how, what's the bowl size?
Need to turn firewood. It wasn't.
What's the bowl size? How big is that sucker? Mr. Ross.
What I've heard, the one might be one of the largest soft maples.
Hey, we read
about it in
the paper. You stole the bees.
At the show.
There's a photo.
What? It sounded like.
It's a family squabble, almost like a nasty road horse. The cemetery owns Sugar Grove, there's Sugar Grove board, there's cemetery board. And somebody came in and just
Back porch.
it's kind of, you never knew what new things she was. Oh, we're gonna do this. No, we're gonna close further. Nobody will know that the gas on it. Yeah, so if I want it out of there before.
Yeah.
I'm not super picky about, well, actually, I am picky about, well, I'm not picky.
What's your name?
I will say that not all boilers are created. It's more of whether they.
You know, who comes to
Gloves get exposed. They get exposed to temperatures. They start to deteriorate.
Volunteer and all kinds.
Passing anybody?
I like them.
Yeah,
They're called vents. They're expensive.
it's more comfortable.
They have time to work by half of it.
Yeah.
Used to be minimums, but, it's just the.
Hey, there was a band.
They're trash, they're kind of box.
Yeah.
But she's always.
It's like a lot of probably, like, it's pretty out. Boxes of mile.
Money, honey? Right, right.
Oh, I don't know. Maybe 15 feet on the other one. 30 feet apart, I don't know.
Yeah, yeah.
Cold day.
He just drove by your house, Dan. Wait.
Oh, yeah.
Where the dog got that.
Not this time. I know.
And so I put the block down.
We've been out for most of the time, so usually I get home from work.
The one I got, I'm on the other. So I'm gonna do more.
But you know. Because, you know,
You get your plans for your solar.
It's nice.
The internet. Having a couple water, we got it. So,
In Detroit, I should be we've seen them and across the road in the tree farm over there. It's more than up here. Frame at the bottom.
lately, I've been around the edge of it. They found that it's fine, it's intentional, I don't know. I didn't know the dog was there. I know about that. And everything. I haven't seen any from our house.
Inside.
Stick over here by you.
Almost have Thanksgiving, didn't need us to help.
Right on the other side of the room.
I've seen it, it's all like this.
You're not doing it. That's the one. That was pretty good.
Yeah, we never get back in school.
Incidentally, in Menards, that's when I want to go to. So you talk.
Yeah, that's got all the.
Nice seating today. We got a lot of dinners taken up. Hey, Dr. Nager, can you hear me?
Yes.
Hey, I just sent you a message on Zoom. I think I neglected to get a bio from you so I could properly introduce you. If you could send me a paragraph or whatever you got going on, I can definitely introduce you to the group when the time comes.
Email it to you?
Yeah, that'll work. Yep.
Okay, cool.
Thanks so much. Appreciate it.
Okay. Yeah, it's one of those that'll be really lovely. Or I haven't eaten too. If I need. Yeah, no, I thought.
I haven't heard back.
Did you sign in, girls? Yeah.
Thursday.
That's she first. How are you, kid? Thanks so much for coming. Oh, I did.
Yeah, I bought it and I mailed it. Okay.
I try not to have that one big.
Dan, are you gonna be around this weekend?
Oh, little one.
What was,
Yeah, so if you're out in business, jump in. No.
Remind me.
Saturday night. We heard back.
Right here?
And then you do a similar.
Yes.
I can introduce him via. More. Over the gratitude.
Oh, okay. Herbie survived. I knew if I didn't order. Especially during the last few years, so. But then he's still going to.
Typically, we don't have this because it was put together by Nick for the extension. This is just a demographic, so you know, male, female, other ethnicity and stuff like that. So it's just that he needs it for his job. So well, I throw it on there. If you need a pen.
It shows the name of the park director in parentheses that have.
Okay, one of the things, the extension office for employees or programs, through the extension, they have to show that they are meeting the goals of the community. And something else I'm gonna try and start, if you need a pen, let me know.
Oh, I got it.
Okay. I'm gonna start trying, okay, I have a bag here, and it's from Germany. So I'm gonna try and have a monthly tasting of something that most of us may not have. Hello there. You know, so it's you got a chance to taste it. And stuff, so take a look and see what German honey. And I was actually quite impressed with the our club was located in Germany. There was she was over in Germany for 18 years. She knew somebody that was a beekeeper over there, knew she was coming back here, so looked it up and found them. I was impressed. I got a note from somebody in Germany. Yeah, about the club. I thought pretty good. Hello there. Sign on in for me, please.
Sure.
I'm going to give you some demographics.
Yes.
Okay. For those of you who don't know me, I'm PK. I'm the treasurer for the club, kind of the troublemaker. Tom, raise your hand. Which Tom? Please be aware there are two Toms. Tom K. Crest is our president. Tom Panconen, raise your hand, the honey pimp apiary. He's one of our consultants. James, raise your hand. He's on the board. Cynthia, raise your hand. She is the equipment manager and librarian. She is a very important person to know. So when you want to use the equipment, you kind of make the reservations through her. And please be aware that at certain times of the year, it gets a little crazy as far as the extractors. We do have two, a 6-frame and a 4-frame. 6-frame is radial, 4-frame is tangent, but the 4-frame handles deep frames better. The 6 one doesn't handle them well. We've got the hand capping kit and stuff like that. We'll have a demonstration in the fall. Okay, so that's nobody else is here. All right. Okay. I have started our inspections of the hives. The hives are at my place now, and I'm kind of rotating between Fridays, Saturdays, Sundays, and Mondays. So one week will be a Friday, and another week will be a Saturday, another week will be a Sunday, another week will be a Monday. Kind of depends on your schedule. It'll be a little bit of working. We moved some hives around, stabilized some stands, did a few inspections. So it'll be at my house, which is southwest of Hayward. Typically like 11 to 2-ish is the time frame. But you know what? If those dates don't work and you want to come out and just kind of, hey, I want to see what's going on, just let me know and I can make other times too. So I'm flexible like that because again, that hands-on. Lynette, who gave us German honey, she actually brought her 8-year-old with her. And, so he got to, go in the hive a little bit. So again, I'm pretty open to whenever you want. We have 3 hives currently going. I'll be putting up swarm traps here soon. So we've got a little bit of work to be able to do within that. So my next one will be on Monday, the 3rd. So that's when the next one is. I did Sunday, so I'm doing Monday. And I think I go to Friday. Anyhow, I'll be putting those dates out there. Like I said, it's an opportunity to see some different things because sometimes you like, oh, what's going on or and things like that. So I'm more than happy to have you at my place. Let's see what else. We this spring we had a total of, let's see, I think we've got 40 plus more of new beekeepers from the spring classes, which was a good number. Yeah, we did have quite a few come in and stuff like that. I am doing a class next Monday on native bees, and then I'm doing one on pollinator gardens through Heartland. So, what you want to do for your pollinator gardens, planting and things like that, some thoughts on within that I'll be teaching. So, what else do I need to tell them?
So real quick, I see new faces. Who's new? Tell us about yourself a little bit. How'd you wind up here? How'd you find out?
My name is Eric Rosenthal. I went through the beekeeping class not too long ago, and then I was here not last month because I missed it on my calendar, but the month before that.
How did you find us? The class.
I just Facebook, just kind of looking around. My wife is a huge gardener. She has 12 beds, roughly, like her own greenhouse, her own hoop house, and she's like, "Hey, you know what'd be awesome? If we had bees." Hey, you know what I don't have time for?
But she's got a guy.
Do what?
But she's got a guy.
Yeah, exactly.
Somebody.
Or she will here soon. Actually, I just ordered my bees not too long ago. For May and just kind of getting everything set up and hoping it works.
Who else we got that's new?
I
Okay.
guess I'm new, Tom.
You're not new. You've been here a while.
I
Go ahead.
haven't been
Tell-
here,
tell us about
so,
it.
How'd you
Yeah,
find it?
yeah.
I haven't done bees yet. How'd I find you? I think it was Nick here at the extension had come out to our property and told me I needed to take this class if I was interested. Is he in here?
He's hiding in his office. Right next, see, he's waving
Yep.
to you.
Hey, quick sound check. Can everybody hear me okay?
Yes, sir.
All right, great. I'm gonna introduce our speaker when it's time, but thanks for
Okay.
letting me sound check. And yes, that was me. I did visit your property.
Yep,
Okay.
so I'm going to start with a new and a package and kind of compare the two, so. Looking forward to it.
All right.
You wanna tell us, you said your brother got you suckered into it?
Yeah.
Love those brothers.
Started up in Detroit with it, and he told me I need to find something else to do in my spare time, so here I am. See how it works out.
Do you have bees ordered? You're gonna get bees this
No,
year?
I don't have anything. The only thing I got is Beekeepers for Dummies.
So you're gonna figure it out.
Okay, you're, you know, once a week and come play with me.
We're gonna talk about that actually better than presentation.
Okay.
So that'd be perfect then. How about you?
Sharon Bachman, I'm from near Lexington. And I've been with the, I've
I
got
just
these
put two
from
and two
you,
together,
and I've been with the Peoria group, but that's just a long way to travel. And it was, honestly, it was difficult finding how to get hold of somebody from your group. I tried a couple of years ago. And I can't remember now how I got
Okay.
your email, maybe somebody from Peoria.
If you went on the CIBA web, CIBA Gmail,
She was probably a couple of years ago. It was probably right before we changed the website.
yeah, we have to change that and
So
stuff.
I
Yeah.
found you and
So
here I am.
I guess the bottom line for everybody, even if you're old timers or thinking about us or whatever, you're online. Make sure you sign in with PK and she gets your email address and she can read it correctly and say it back to you. She'll get you on her mailing list. And then you'll get an email that says show up to XYZ event. Whether that's to go play with the bees or whether that's when we do bees and brews or whether that's when we have our club meetings. Or any other shenanigans that we got going on.
I
How
got,
about the people online? I see some faces there. Maybe not. Anybody new online?
Ella's been here before, Melissa, I don't know who Wood is. And then Polly's been here before. Nick's there, I'm not sure who Wood is. Hi, Wood.
Dylan's from Peoria.
Let's go ahead and kick off.
We do have eight chat things we might want to check on real quick.
Okay.
Give me a second.
Okay, I'm the one who says asking if you can be helpful. On the 4th, I'm gonna be at ISU's NutriFest. Having a display, they're gonna have a whole series of stuff talking about that, so I'm the one, you know, that kind of goes out and says, hey, you know, come check out the bees. So I'm gonna be doing that if anybody wants to help, so I'll be sending something out to say, can you be helpful for me? So I do, JD's helped me, Ken's helped me. I don't think you've.
Not lately.
Yeah.
Used to help me with something. So I, you know, Cynthia and James did, we were just at Central Catholic, for the, what was that for? That was,
Flower garden.
no, it's, Home Lawn and Garden Days, that's what it was. And stuff, so again, it's just getting the word out there, and, as you see, I'm very shy. So anything else that we need to do, Tom's?
Well, that 4th is coming up awful fast. You want to
Yeah.
give us some more information about that?
Tuesday, it's gonna be 11 to 2. And we're not selling honey because it's on campus and they kind of said no, so it's just talking about bees. Okay, so yeah, it's kind of, it's a last minute kind of, like, okay, we'll do it. You know me, I'll be there, how's that?
Okay.
Okay, all right, Nick, I'm gonna turn it over to you.
All right, excellent. So everybody can hear me once again? Sound coming through okay? Don't need to turn it up or anything?
Yeah,
All right,
okay,
wonderful.
Very good.
All right, I'm gonna go ahead and introduce our speaker. And but first, I don't know if anybody was here last February when I did a talk on honeybees being seen foraging mushroom compounds from wild species of mushroom. Anybody quick raise of hands in the program for that one? Okay, cool. Yeah, we can't see everybody through the MondoPad view of the room. So sorry about that, Dr. Nager and everybody else online. But yeah, so the paper that I presented, among others, to the group last year, just to refresh everybody's memory, was titled Extracts of Polypore Mushroom Mycelia Reduce Viruses in Honeybees. And that, among others, was presented at my talk last year to CIBA, and it was really interesting stuff. And I'm pleased to bring our speaker to CIBA tonight, all the way from Washington State University, Dr. Nick Nager. He is one of the authors of this paper that I presented last year. So I personally am thrilled to hear his talk. And so here is his intro that he has so kindly provided me. Dr. Nick Nager is an entomologist and geneticist that has been researching honeybees for over half his life. Before joining the WSU team, his doctoral work at the University of Illinois Urbana-Champaign focused on the genetics of behavior, where he documented how networks in genes and honeybee, oh, there's something in the way here, where genes in the honeybee brain turn on and off foraging or mating behavior. His current research seeks to find innovative ways to improve honeybee health. This includes using fungi to create better bee feed additives and using pathogenic fungi to kill varroa mites. So really excited to hear his talk, and without further ado, Dr. Nager.
All right, well, thank you for the introduction. I'll go ahead and share screen now. And hopefully you can see my mouse also.
Yep.
All right, well, yeah, thank you for the introduction. It's a bit fun to talk to this group. I did my PhD at University of Illinois. And so when I even heard earlier the home lawn and garden days, like I gave a talk there in like 2012. So kind of a bit of fun to be back, if only on Zoom. Well, anyway, most of my research now is dealing with fungi, kind of the forgotten about kingdom of life, and honeybees. There are two main projects that I work on, these two main branches of research, and they are fairly separate, although both of them use fungi. The first one that I will talk about is using fungi to create honeybee feeds. And then the other one is about using fungi to kill varroa, because we need more dead varroa in the world. All of this is in collaboration with Dr. Steve Shepard and Dr. Jennifer Hahn also. All right, well, to kind of frame the talk, what I'd like to point out is that bees do forage on more than flowers. A lot of beekeepers are very aware about bees foraging on tree sap. They bring it back to the hive and turn it into propolis. And this tree sap contains a lot of good antimicrobial compounds that can help bees fight chalk brood or other infections like that. The tree doesn't have antibodies. It uses chemistry for its immune system, and the bees can simply bring that sap back to the hive and benefit from that. Bees will also forage on honeydew. This is a sugary secretion that comes out of the back end of aphids and other sap-sucking insects, and bees will forage on that. There are multiple documented instances of bees foraging on algae, and it looks like they get vitamins and minerals from it. And in fact, many beekeepers have noticed that they will put out clean water for their bees, only to have the bees prefer a scuzzy pond. Bees kind of like their algae too. And then you will occasionally see bees foraging on mud or sometimes manure. I actually saw manure foraging in Illinois in the late summer when there would be a kind of a drought and then a rain where you get little, where the cow patties would be dry and then rain would collect in little puddles. And it looks like that leaches out a little bit of mineral from the manure or mud or whatever, and then the bees can drink that up as a way of getting some calcium, iron, a few other things like that. Well, bees also forage on fungi. This is getting to be appreciated more and more. So here's some various photos that I've collected over the years. Most of the time they forage on wood decay fungi, as opposed to like leaf decay or like various molds or so on. They tend to like wood decay fungi, although down here on the bottom right is a bee chowing down on a member of the bolete family. So that's the mushrooms that give us porcini mushrooms. So that bee might be having a gourmet meal, but most of them focus on this wood decay fungi. The story of this research really starts with this man here, mycologist Paul Stamets. He lives here in Washington on the west side of the state where it's nice and rainy and perfect mushroom habitat. And he had all of these cultivated mushroom beds, and over, bees would show up and start foraging in these mushroom beds. Now, what Paul noticed was that they weren't really eating the mushroom mycelium or any parts of the physical mushroom body, but instead they tended to drink what we call fungal exudate. And so this exudate is like sweat that comes off of mushrooms and mushroom mycelium. It contains a lot of different things in it. This is their only way of getting what's inside their body on the outside. So it could be to attract flies to carry spores away. It could be to to have more noxious chemicals to deter beetles that could be eating it. It could be metabolic byproducts that the mushroom's just trying to get away. If it has a bunch of extra calcium or sodium or so on, it can get rid of it this way. So anyway, these exudates contain many, many different things. It's a very complicated mixture, but for a lot of different species of fungi, bees tend to like foraging on it. In particular, Paul noticed that the wood decay fungi of groups that were used in traditional Eastern medicines, traditional Chinese medicine, that some of those were particularly popular. And these mushrooms have been used as, basic health treatments for, millennia. And now that we have some of them in the lab, we're finding that there is a little bit too, that they have some level of antiviral property, some level of anti-tumor properties, and so on. Generally some good things. So when Paul saw that bees were having problems with viruses, he thought, well, is it crazy to think that bees are self-medicating on these mushrooms? The answer is no, it's really not crazy. I don't want to get into it too much, but there are actually many, many different examples of insects self-medicating, usually choosing particularly weird or toxic food on purpose to rid themselves of gut parasites or things like that. So this is something that insects can do. And among insects, bees are pretty smart. So the possibility is there. Well, anyway, to kind of do this research, what we decided to do was just start feeding bees extracts from mushrooms and see what happens. And amazingly, bees in cages that get these mushroom extracts in their sugar water live longer. Overall, what happened was Paul's company, Fungi Perfecti, we have them grow different species of mushrooms, sometimes on different wood substrates, different variations like that. And then they do these extractions and send us these different things to test. And then we can feed it to bees in cages and see how it affects their health. One of the main things we look at is lifespan. Bees only live a few weeks anyway as adults, so we can look at their lifespan in just an experiment over a month. And so here's a survivorship curve. And as bees die off, the curve kind of goes down. Here was an early trial where we were looking at a lot of different species, and this was Ganoderma lucidum is what bees did the best on. And then there's this fomies mushroom, the Laricifomes, and this Inonotus one. And then birch was just the wood we grew it on. We made a tea extract from the birch, and sugar water did the worst in that trial. Over here, we focused on those Ganoderma mushrooms, and we tried Ganoderma lucidum and Ganoderma resinaceum, and they both performed great. This was an amino acid supplementation that I did for scientific reasons. And then honey and sugar. So this is kind of remarkable that these fungal extract augmented sugar syrups actually seem slightly better for the bees than their natural food, honey. Now, the reason for this is probably because of things like nosema spores in honey. And so when the bees are in cages and they end up ingesting spores, it doesn't do them a lot of good. But overall, with sugar water and extracts, we can give them a sterile food, and it does end up benefiting them in this caged situation. Where we've gotten the most press about this work is that feeding these extracts to the bees in the cages, it seems to help them fight viruses. And we've tested deformed wing virus, Lake Sinai virus. We also tested a gut parasite called Lutmeria. And in each of these cases, it looks like feeding bees these extracts helps them lower these various viruses or gut parasites. The bees do seem healthier overall. These are the data from the paper that Nick mentioned in the intro. Lower bars means less viruses. And what we saw here was a dose response. So low dose, high dose, low dose, high dose. The higher the dose, we got more reduction in viruses. So things look really good. The bees seem healthier, and that's great. But there's a big question as to why, like what's going on. And so one of the things is, well, bees are mushrooms, that's food. Is it as simple as nutrition? Bees need carbohydrates for energy. They need proteins and amino acids, essential fatty acids, vitamins, minerals. That's a basic set. That's not too different from what you or I need. But bees must get everything from flowers or maybe some of those other environmental sources that I talked about earlier. Well, one of the things we did is we took these extracts and we sent it off for nutritional testing just like you would with any human food, and just ask what is in here. And the first thing that stood out was the minerals. So there's a fair number of minerals in these extracts. And when we look at the ratios are really close to what we find in the bee's natural diet. One thing of note, kind of a difference between bees and humans, is bees want a lot of potassium and very little sodium in their diet over here. You know, we humans want a lot more sodium, less potassium, a little more phosphorus, a little less magnesium. It's not quite the same. But anyway, the mineral concentrations that are in these extracts end up being very close to what the bees get in their natural diet. And that could be part of what's going on. They also get some B vitamins as part of it. So there could be nutrition as part of the picture of what's going on. But overall, we did not expect nutrition to help bees fight viruses, really. And so we think there's more going on than that. Other types of nutrients that we've kind of looked at to some degree, one of them is sterols. You know, we need some sterols in our body. We need some cholesterol. We use them to make hormones and so on, and bees do similarly. Here is a bee hormone that's very important for coordinating when they pupate and emerge as an adult, and it's made from the sterol backbone. Well, bees cannot make it on their own. They have to get it in their diet, and fungi is one of the places they can get it. Fungi produce high concentrations of sterols in their cell membranes to help make their cell membranes stiff. And as it ends up, nutritionally, many of you have probably been feeding sterols from yeasts, and yeasts are unicellular fungi. They're just fungi that don't go into multicellular organisms. Those yeasts have sterols, and only recently, a lab at Oregon State has shown that these sterols are really important for bee health. So a little bit of brewer's yeast in your pollen patty is a good thing. You get more than vitamins and protein from that. And we think that now we have a liquid feed that can deliver a similar thing. Another thing I want to highlight is these phenolic compounds, sometimes called polyphenols. This is a diverse group of compounds that are found in nectars and pollens, and they're also really common in fruits and vegetables. So May Berenbaum over here, University of Illinois, not too far, her lab has shown that these compounds upregulate the bee immune system, particularly their detoxification system, so they can handle pesticides better, and then also some genes associated with pathogen fighting. To get into this a little bit deeper, these polyphenols, what are they? Well, most of them have this antioxidant activity. If you hear about the good compound from red wine, well, that's one of these, or, you know, compound from blueberries that they transfect into tomatoes. You know, these good for you compounds, a lot of them are these phenolic ones. They contribute to the flavor of foods and then also the colors. They are what makes your blueberry blue and your raspberry red. Generally, most health professionals think that a mix of different phenolic compounds is what's best for you. So even though there are individual ones that are really good, a mix of them is what's best. And what I find fascinating is our own experience of just eating and tasting food confirms that. When you go to a place that sells fruit smoothies, they do not sell just strawberries. We like a mix of these different phenolic compounds in our diets, and bees do too. That's one of the reasons why they really like to forage on a diversity of flowers, is they get a diversity of these different phenolic compounds. And that way they're getting, well, a diversity of diet. And they get a fruit salad rather than having to have a meal of entirely blueberries, would be the analogy. So one of the things that we have found is that when fungi degrade wood, and if you remember, bees tended to go for wood decay fungi, the degradation of the wood compound lignin results in all of these different polyphenolic compounds. And so what I think is happening is, just as fungi are degrading wood, the byproducts of that metabolism happens to create compounds that are naturally found in nectars and pollens and are quite good for bees. So this is another part of what we think is going on. And over here, diversity of phenolic compounds found in honeys. And then here are my different fungal extracts showing similar shades of brown from all of these different types of phenolics mixed together. One of the main things my lab is trying to get into is how exactly these nutritional things interact with the immune system of the bee. So bees have a different immune system from you and I. And one of the tricks that they can do, they cannot do antibodies, but they have other tricks. One of them is pictured over here. If you look, there's an invader, and this invading microbe has been swarmed by cells. And these cells are now depositing a black layer onto it. This layer is melanin. It's the same melanin that's we use to darken our skin. But when bees produce it as their immune system, they do it like plastic. It's like a sheet of plastic wrap that they can wrap around a bacteria, a virus, a parasite, whatever they want, whatever they realize is not supposed to be there. They just wrap it up in this plastic until it suffocates. And this thing can't eat or breathe or do anything. And then the immune system can slowly chew it up and get it out of the body over time. Well, anyway, we've discovered that dietary phenolic compounds are actually really good and help boost this melanization aspect of the bee immune system. So I have a master's student that's working on that right now. All right, well, to kind of close this first part, we're working to get it into beekeepers' hands. And this is a bit new because there are not a lot of registered products on the market. And working with a university and then also working with private industry that doesn't want to get sued, we definitely want to do everything above board and with government regulation. So we're going through the FDA, Food and Drug Administration. Bees, whatever bees eat goes into honey, which goes into the human food chain. So FDA is very interested. Bees are considered livestock. Same as cattle, you are eating the downstream products. We're also working with the American Association of Feed Control Officials. They are the ones that like standardize dog food or cattle feed, give you that nice nutritional label so you know exactly what's in it. We're working with them to make sure that this is a standardized product. That's kind of the main path. What I have here is pointing out that even though I talked about these sterols and these phenolic compounds and all these interesting things that are good for bees, when it comes to this sort of government regulation, all they want to know is calories, protein. You're making a feed, right? And so even though we eat things like broccoli for more than its calories, but the nutrition label for broccoli has like calories, protein, and vitamin C, that's what's going to happen here. It's going to be marketed as a basic fungal-based bee feed that contains vitamins and minerals, but hopefully most people know that there's more than that in there too. All right, hopefully we'll just kind of save questions for the end. And what I like to do is transition to the second part here, which is about using fungi to kill varroa mites, because everybody loves a dead mite. Well, fungi are pretty good pathogens of insects. They do really well in cold-blooded organisms that lack the ability to create antibodies. So, we have more problems with things like viruses. Insects tend to have problems with fungi. Well, what we did was we keyed in on this. There's a couple different fungi that can infect and kill several different types of mites or other soft-bodied creatures. Basically, we landed on this fungus called Metarhizium. It is this entomopathogenic fungus, which means it kills insects. It is native. It's found in soils across the US and throughout the world, actually, very common in agricultural soils. And when we did a little fungal lab as part of mycology class at the local community garden, the second most common fungus in that soil was Metarhizium. It's really common where it attacks and kills soil invertebrates. It's very safe for human. You can spray it in kitchens and schools and things like that. It tends to be a little expensive though, and so doesn't get used a whole lot in large scale applications. It can kill many species of mites and other soft-bodied things, but honeybees seem largely resistant to it. If we crank up the dose, it will make them sick, but they seem very resistant to it in a great way. So one of the things we identified with this fungus, we knew it would kill mites, but there's this problem with it in that here, if we have a fungus stored at a refrigerator temperature and then germinated at room temperature, it's kind of happy. These bits growing out of it are like roots. This is what searches for a weak point into the mite to kill. It's kind of what we want to see. If we store the spores at a beehive temperature, 35 degrees Celsius, and then germinate them at room temperature, most of the spores have died, and what is there is a little bit stunted. If we try to germinate the spores at beehive temperatures, even if they were healthy before, they are fairly stunted. And if we store them in beehive temperatures and then germinate them in beehive temperatures, almost everything is dead, and what is there is weak. So we have a fungus that can kill mites, but you can't put it in beehives. What good is that? Well, anyway, Dr. Jennifer Hahn and I sat down, did a lot of reading, and decided that we could select and create new strains from this starting point. This is a basic diagram of our selection regime. Basically, we use laboratory stressors, laboratory incubators, and everything we could do to speed up generation times to evolve this fungus as fast as possible to be able to tolerate temperatures. And then we follow this up with field tests to make sure it could still kill Varroa. Ultimately, what we wanted to do was take this soil fungus that can kill several different mites and so on, and select for it into a very, very specific form, much like how one ancestor brassica plant was selected into cabbage, into Brussels sprouts, into kohlrabi, and into kale. We think we can take this fungus and select for it to kill mites and mites only, or at least mites very specifically, and leave bees alone. This was a huge effort. We screened something like 36,000 mites for what killed them. And so that was definitely not a trivial amount of effort. Jennifer Hahn and I spent many, many days working 8 a.m. to 9 a.m. doing these autopsies on mites. And I joked that by the end of the years of work, we were either going to be married or kill each other. And we are married now. So anyway, after our thermotolerance selection, things, we knew the fungus could then grow in temperatures, but it might have lost other things during evolution. And so we took it back to the field and began to repeatedly put it through mites. Then the mites that were killed from the fungus, we grew fungus out of it and then deliver it to the hive again. And so we're just doing this repeated selection through mites, and we are specifically choosing strains that when we put just a little bit of spores in the hive, it kills the highest number of mites. Here's what we're seeing with these generations of selection. And early on, most of what we did to this fungus in the lab did not benefit it when it was back out trying to kill mites. And so initially we had just like a tiny bit where only 5% of the mites in the hive were killed. However, after just one generation passed through the mite, we immediately got that back up to killing 50% of the mites. And then here's where things get more interesting. The thermotolerant selection finally begins to shine through. So rather than our treatments lasting 7 to 9 days, here it began to trickle onto 13 to 18 days. And then here we actually have it lasting several months. So one treatment now is killing about 5 times more mites than it did initially because of the selection that we've done. So that is fantastic. Here we try to do colony longevity. If we were just treating with nothing but this fungus, can we keep the colonies alive? The answer was yes, until our control colonies turned into mite bombs and everything died. So if any beekeepers are not familiar with this, when a hive dies from Varroa, everything nearby gets massive loads of Varroa and dies. So do not be that beekeeper. Do not let your hives die from Varroa or all your neighbors will die too. All right, so overall, we've basically found out that this new strain is much, much happier in beehive temperatures. We get better germination, a longer germination tube, it kills more mites, things look pretty good. However, we have to figure out how to deliver it to the hive. We were doing a lot with Petri dishes just on top of the hive, but that is not a way to mass manufacture or deliver this. So we've been trying to work with these different delivery methods. Here's one where we use these fiber bags that the bees could chew through, and the grain that we grew the fungus on then would slowly spill out over the hive. That worked out really well. It outperformed an oxalic acid control treatment that we had. And so again, we're getting some level of control. It looks like it mainly hits the mites that are not in the brood, right? It mainly hits the ones on the bees rather than under the capping. And so we might have to do multiple treatments, much like an oxalic acid drip. But it seems to be killing mites at about that level. And it seems to be very safe for bees in a very important way. We've done a lot of work on how to grow this. And so here is a different growth substrate test. I don't want to take you through it, but over here, rice and then pearled barley were the two winners there. It is remarkable. The husk on the barley prevented growth. We had to have pearled barley to get that to work. As it ends up, rice is the cheapest. We can get organic rice that is very low on fungicides, which is good for growing fungus. We're moving forward with that. And we're kind of ramping it up to bigger grow systems where we can grow kilos of rice and then billions and billions of spores in one go. We're continuing to work with delivery methods. Our little mesh bags worked okay, but when hives get weak, they stopped working it, stopped cleaning it out, stopped interacting with it, and then the treatment effect went away. So we need the bees to work it a little bit better. We're doing different kinds of grease patties, wax paper bags, things like that, that bees will chew away and release the spores over time. We're working on all that now. We got some good headway last year, but we don't yet have something that we're comfortable approaching the EPA as like, here's our final product, here's how beekeepers will use it, register this, and we'll move forward. So we're almost there, but not quite. So we're working with the Office of Commercialization. With it being a new strain, we're trying to get some level of patenting on it so we can control it also. This is a living biological thing. And, there are some movies out there about entomopathogenic fungi going crazy and causing zombies and all that. I don't think that's going to happen whatsoever, but patenting gives us control in a way that I like, especially until we know more about the overall effects of this fungus. So anyway, we're moving forward with commercialization and patenting. More recently, I found a company that does seem willing to license this technology and begin to grow it in mass, as long as we're willing to help guide them how to do so. And so getting this registered as a new pesticide is going to be a multi-year process. Because we have to show human safety and a lot of additional things, but it's moving forward. And I'm very hopeful we will have a new tool to kill Varroa that is very safe for bees. And even if it's not as strong as something like Amitraz, and you might still need to do stronger mite treatments in the fall, I still think it's going to be valuable to just help keep mite populations lower. And then when you do have to treat with harder chemicals, less frequently then, in a way that will be good for bees. So just to kind of end, here's kind of a final thing. Most people like to watch Varroa die. So here's some mites that we dipped in spores and then stuck them to the Petri dish. And here you see mycelium, fungal hyphae growing out of it. And then here in a bit, the greenish color, the brownish green is spores being produced. So I always like to watch mites die. That's always good to see. And with that, I'll take any questions.
Is it temperature affected?
I'm having some trouble hearing the room. So if somebody could move closer to the microphone or have somebody relay it to me.
Work at lower temperatures than.
He says, does it work at lower temperatures?
Yes, it appears to. So we did one experiment with trying to get mites off of overwintering bees, but that meant leaving mites on overwintering bees, and most of those colonies died regardless of our treatment. If you leave mites on your bees going into winter, even if you kill those mites, the viruses are going to persist and those bees will die anyway. So it does work at cooler temperatures. And that makes it good for the fall treatments and so on. But really the higher temperatures is what we were battling. But it will still work at the cooler temperatures. And then we get a longer treatment duration in September than we do in August.
how long do you expect it before you can get approval for it, I'll ask?
I would love to be super optimistic and say a year or two, but I think we're looking more at 3 to 4. Getting a new pesticide to the market is difficult. And, just here's one of the things to let you know, like how complicated and how big it is to get a new pesticide. Even after we do our own bee safety data, our own human safety data, we get our entire package together and go to the federal government and say, here's our package to approve to get this as an EPA registered pesticide. That comes with an $800,000 price tag to have them do all of their reviews also. I mean, it's really a massive bureaucratic thing, kind of set up for the Monsantos of the world. I see routes that we can get this done with our smaller groups. There are certain pathways in the regulation. I don't want to get into it too much, but there are things like Section 18 emergency approval. And if bees are dying, that might be an emergency, and we might be able to not provide a full safety packet before use at your own risk is approved. So again, maybe 2 years, that's kind of the near end. I think it's gonna be more like 3. And my experience with science says it's gonna be 4 because things don't go right the first time. But I'm still optimistic overall. And the other thing that I haven't pointed out is, as a proof of concept, we have shown that these fungi are changeable in a better way. We can make them better. And so they're not being used a whole lot because, sometimes if the sun is too bright, it'll kill the spores, or the temperature is too hot, or these many other things, or, maybe it's good at this pest, but not that pest. We've really shown that these things are evolvable. We can change them. And this gives me great hope that others will pick this up and say, maybe we can spray a nice safe fungus on things like fruits and vegetables rather than chemicals. So hopefully this is kind of the beginning of a broader movement towards using these much, much safer biologically based pesticides rather than chemically based pesticides.
So we have 2 other questions. One is, I'll give you both questions. You can answer them if that's all right.
Yeah.
The one is, will you be able to use this with honey supers on? And the other one is, will the spores impact things like small hive beetles or other hive pests?
All right, so in terms of supers, currently not a lot. There's not a lot of mite treatments you can use with supers on. Hop Guard was one of them. And then I believe the recent, maybe I shouldn't say this. I thought the oxalic acid label changed this year. Check me on this. You might be able to do certain oxalic acid treatments with honey supers on as you're making honey. I would say there is a good chance that this would be one that we can get through because the human safety element is established. It does seem so safe for mammals in general that it seems likely if there was only a commercial honey industry, most of what you get at the grocery store has been highly filtered to remove pollen grains and things like that. And that prevents crystallization and gives it a longer shelf life. I believe that those industrial processes actually remove and kill whatever spores are there. And so even as some get into the honey, I think it's going to be removed and be safe with most commercial processing. But we, of course, want something that we wanted to, if it's going to be approved for honey supers, it's going to be approved for raw backyard honey production also. And so I hope we can get that. That might not come at the very beginning, but yeah, it would be really amazing to get another varroa control tool that we can use during the honey flow. Particularly, it's not as bad in Illinois, but here in Washington, I'm actually between mountain ranges in a rain shadow. we actually get less than half the rain that central Illinois does. It's really quite dry. And so we have these springtime rains, but our season of honey production is extremely compressed. And it's at a time when the hives are growing and so on. Now, most of the time the hives are outgrowing their problems at that point, but our beekeeping season, when we can like act on those bees and like work with them, is very compressed. And yeah, I'd love to have more things that we can do to make sure that mites are in control when the season starts. Because it feels like here we're in winter until April, May starts, the bees start to build up, we produce 70, 80 pounds of honey per hive, no problem during June. And then the flow's done mid-July. And we have to start feeding sugar in August, September, October, November. It's really quite miserable having no late summer or fall flowers. The other thing was a question about small hive beetle. So I had some hope for this. Another one of my annoyances, because with the way university systems work, we have a lot of undergraduate help, and basically things like wax moth that are generally easy to control, we have outbreaks of because people aren't good at people put contaminated comb back in the main storage room is what it comes down to. So we always have wax moth problems. And so I took our selected strain and tried to use it on wax moth, and it seemed to do extremely poorly, worse than our starting strain. And I think as we select so heavily for it to kill mites, its ability to kill these other things is going down. And so I don't have a lot of evidence from just my one little trial with wax moth, but I think we're really making a mite-specific pesticide, and I don't think it's going to hit these other things. Now, we can certainly try to either with this strain or start over with a different beetle-specific one. There are definitely options like that. There are strains of Metarhizium that are known that only affect grasshoppers. And so when they have locust outbreaks in Africa, they can spray that. So the possibilities exist, but probably not with this strain. I think hive beetles will be a different issue. Okay, I feel like it took a lot of time. Are there any other questions?
I got one. The fungal extracts you were talking about earlier, are those from one specific mushroom type, or are those from like numerous different types of mushrooms?
Yep. So overall, I would say I tested 5 different clades of mushrooms. The polypores as an order really stood out. All right, so then within the polypores, which there's a bunch of them, these are the bracket fungi, shelf fungi. These are the ones that grow on trees and they have pores instead of gills on the bottom. So the polypores did really well. And I would say of those, about half did something positive. I did find, actually our most common local wild polypore was slightly toxic to bees. So, you know, it's not all of them. But then within them, the group that really shined was the reishi mushrooms in the genus Ganoderma. And these are really quite common. Many different companies are growing these to make different teas and so on. You know, you can get dried powdered Ganoderma mushrooms in tea bags, make your own little medicinal mushroom tea. These, you know, quite common. I would say Ganoderma is the only one that we have shown repeatedly to be good and safe. Some of the other ones, there's a really cool mushroom called the agaricon, and it seemed to be very dependent on the bees. Sometimes it seemed good for them, sometimes it seemed kind of bad for them. But these Ganoderma ones are the ones that are kind of testing out over and over to be quite good. And I've worked with Fungi Perfecti as they've been making these to kind of tweak some of the production in a way that we get minerals and some of these extra nutritional things in it. So yeah, overall, I would say we know wood decay mushrooms are better. The polypores with them is a pretty good group. And then within those, the reishi group or the Ganodermas, same thing. Those are the good ones. And actually, this has been, so yeah, here, let's see. Here's a human Ganoderma one being marketed. that little red mushroom with the white fringe on it is actually very similar to one of the species we're using. So they sell these for human consumption too, but there's a difference that the fungus normally likes to grow on wood, and for the bees, we're growing it on wood, but you can't sell wood to humans for food consumption or so on. So all the human products have to be grown on rice or something edible.
Man.
How is your research funded?
Oh, what a fascinating question. Yeah, so it's been a mix. I would say what's really made this work was 2 major foundations that gave money to support it. And then what I did was I took those monies and kind of hardline that into the fungal extract research, hardline that into my own salary, and then I write other grants to kind of support things overall. So when I do like beekeeping workshops and so on, I have like a specialty crop block grant to basically do bee education and bee health things. And then that pays like 15% of my salary. And when I get that 15%, then I can put that back into the fungal research. So I'm actually juggling quite a few different things, but the backbone of it that keeps it all running is the Jonathan and Kathleen Altman Foundation. And before that, it was the Whittier Foundation. Whittier Foundation the Whittiers made a lot of money from petroleum, and now they fund a lot of eco-friendly projects. And we are one of them, so.
Well, that is a super fascinating topic and talk, Dr. Nager, and really great follow-up questions from the audience as well. I know that we have other plans for this evening's meeting, so everybody, let's give Dr. Nager a round of applause for an excellent presentation.
All right, thanks everyone.
Thanks a lot. Maybe I'll send you a follow-up question or two. That was awesome. Thanks again.
Sounds good.
Great, thank you. Now, our follow-up is how to grow your own mushrooms. Okay, so Gabe and Brenna. Okay, they have a company called Totally Normal Fungus, right? And they can show you how to grow fungus and how you use beeswax in this growing process. How long have you guys been doing this?
So I personally have been doing this for years. We have been doing this as Totally Normal Fungus, a company, since 2022. So we're a relatively new company.
Okay.
We're still within our first rolling calendar year.
Okay.
Maybe you could do that like right there so the camera picks you up.
I sure can.
One doesn't spin every now and then. Come back here.
Hey, watch it.
Yeah.
And you see,
All right, well, like PK said, my name is Gabe. I am founder of Totally Normal Fungus. This is my business, one of my business partners, Brenna Long. We are a new fungus company that really teaches people how to engage in home mycology. So we don't really produce mushrooms that you can buy from us. We instead provide you the tools to be able to grow your own mushrooms at home. One of the varieties is Ganoderma, aka Ricky, the ones that we were just talking about. So great intro, Dr. Dave. You know, great. So yeah, where do I start? So what I'm showing right now is a low tech or a something that doesn't require a lot of equipment on your end, way to grow mushrooms outside in a controlled environment in your yard. I used this is a log from a crabapple tree that was down last November. It doesn't really matter like really when you harvest the log too much, but the closer you can get to winter, the better, because all of the nutrition that the tree has produced through photosynthesis during the summer is stored in the tree, getting ready to hibernate for the winter. So there is a higher nutritional value for the fungus in the tree if you harvest it kind of like later in the fall, winter. Once they start to bud around, it's a terrible time to cut out a tree for mushrooms. Don't do it, please. But if a tree falls and you're harvesting the wood anyway, if there's a storm or a branch falls, harvest it. It's gonna work. The way we do it at Totally Normal Fungus is pretty simple. We actually provide you one of the ways we can do it. One of the ways that we advertise is we sell you the pre-inoculated mushroom variety for your log. Or if you want to become an extreme hobbyist, like some of us in the room, we also sell you things like liquid cultures, your own mycology jars, things like that, that you can then make your own plugs or get really as far into the hobby as you want, right? So really kicking myself in the rear. So I had a couple of extra plugs that I was going to bring to actually show you. They're really nothing. The plugs themselves aren't really that special. They are untreated wood dowels. The reason it's important to use untreated dowels in this process is to make sure they don't have any fungicides on them. We're trying to grow fungus on the wood, not to repel it, right? After that, we really introduced a liquid cultured variety of a suspended mycelium that's in a liquid culture that we have in a syringe. We introduce it into the container that is that has our plugs, and then it does its thing. After they are fully inoculated and colonized, we just drill holes the depth of the plug into your log. You pound them in, and then after you plug it where you coat it with beeswax, right? This is just, it looks kind of like nice rendered beeswax. It's not. There's a bunch of detritus and gross wood stuff in the bottom. Doesn't need to be clean necessarily. The reason we're putting beeswax over the plugs is to make sure that we're not introducing any additional infection. So what we've done is we've stripped the outer layer of wood from its natural protective layer, the bark, and the inner sapwood of like the nutritious part of the wood is now exposed. So what we want to do is we're plugging it with mushroom mycelium, so that way that mycelium is able to access the nutritional part of the wood, and then we're sealing it to make sure that no other micro like biota or microfauna are able to infect the hole before it's done colonizing. And then you just kind of let it sit. You put it in a nice shady spot of your yard, north side of a fence, north side of your house, next to a tree, whatever. And then it will, within I hate giving out time frames, but usually within 6 months to 2 years. I know it's a very wide time frame, but like Dr. Nick was saying, it is a living creature, and a lot of the colonization rates depend on what the year was like. You know, was it wet, was it dry, was it hot, was it cold? So it's gonna fruit when it's ready to fruit, but the log itself is now inoculated, and then you just kind of wait. So long story short, that is how you do mushroom logs. So are there any questions? There's gonna be a time.
Does it matter what kind of wood? Like,
Yes and no. So you wanna, any sort of deciduous hardwood will work. You wanna avoid things like pine and cypress and things like that. They have natural antifungal properties. There are ways you can use those types of woods for certain varieties of mushrooms, but generally they should be. Yeah, yes.
Once the log starts to fruit, how long will it fruit?
Years, probably about 3 to 5 years. If you harvest them, if they're optimal time, you're probably looking at 5+ years. And a lot of it is, again, variable, like how much did the mushroom produce? Because what's going to happen here is you're introducing what is a saprophytic variety of mushroom, which is a decomposer variety into a log, and it's going to turn into dirt, right? So over a period of time, that log is just going to become a pile of mulch, and once that happens, it's just going to stop hurting, you know. So during that period of time, if it had a bunch of really super heavy flushes and it's expended a lot of energy, it's going to have produced more mushrooms in a shorter period of time than if it just had light flushes and, you know, didn't use all the energy of the log, it would then probably last much longer. So it's variable, but years. So,
So if a guy's producing a lot of sawdust.
yeah.
I, you know, throw out bags from my dust extractor, they're like big bags like that.
Yeah.
And can you introduce that into
Sure.
sawdust?
So not in the way that I just described, but they're, like I said, a lot of the varieties that we have are sacrificed, so they eat wood. And depending on the variety, some of them respond really well to sawdust. As long as you hydrate it to what's called field capacity, which is the point where it holds as much water as it can without being drippy, is really what that means. So really great way to test that is to pick up your sawdust and squeeze it. If it drips, it's too wet. If it doesn't drip, perfect, right? So you want to be wet, not drippy.
And that would be grown in jars more like this or in bags rather than in a pile or something.
Right, so you would have to pasteurize, like there's a process to it. You have to pasteurize it and make sure it's a hospitable environment for the fungus that you're going to introduce to it.
How would
And
you
then,
pasteurize it?
so pasteurization can be done with hot water a lot of the times. So at Totally Normal Fungus, we use a lot of pelletized materials, and that helps because the pelletization process, both with the heat and pressure, kind of pasteurizes anyway. That introducing additional hot water into that gives you a very, very clean substrate. If you're using just raw sawdust, you can do the same thing. Make sure your water is very, very, very hot. There is a technique it's called pillowcasing. It's exactly what it sounds like. You take a pillowcase and you light a bucket, put your very, very, very hot water and your sawdust in the bucket, let it sit for at least 12 hours, take it out, let it drain. Once it stops dripping, you have field capacity sawdust. And then you can introduce something like a grain spawn into it, and depending on your variety, or will not fruit. So you want to introduce a
If
variety
it's
like
in a jar, then how's it fruit?
so it wouldn't actually fruit in the jar, so this is spawn. So this is grain spawn. So you would open it up, you would break it up, and you would introduce it into your sawdust.
Yeah.
And you only really need about 10% spawn by weight compared to sawdust. So it's a pretty low amount. This would colonize gallons. And then you just kind of do it that way. There are techniques to do what's called a bucket grow. That's a lot of times what I recommend for people that don't have a lot of mushroom experience, but are interested in kind of testing it out. So you take a 5-gallon bucket, drill a bunch of holes in it, pasteurize substrate, grain spawn. Then you get mushrooms, right?
These mushrooms are what people would want to eat then.
Yeah, no, these are blue oyster mushrooms. I don't know if people have heard of oyster mushrooms, but they're pretty common, forageable, and they're starting to become a little bit more common in the grocery store, along with lion's mane and things like that. But all of the mushrooms we provided Totally Normal Fungus are ones that are either edible or are medicinal or functional, like the reishi. A lot of people do make teas out of them for personal wellness and health. And apparently bees do too. So that's pretty cool, right? Learned that today. Yeah.
Do you have to roll the log every so often if you've got plugs on the bottom or? No,
You just
so
leave it.
just to get the mushrooms off, you just,
The mushrooms will fruit normally out of the sides and not so close to the ground, but when they do, like fruit really, really close to the ground, they're gonna grow upward and away from the actual earth. So like, it's not gonna hurt anything. There are actually certain varieties that you can do in a log form that are beneficial to bury in the ground, like chicken of the woods, hen of the woods are great examples. And in that situation, you would just plug it from the ground, just like a regular.
we do shiitake.
Okay, yeah.
So I just wondered if we needed to turn it, and I noticed when I was out there that there were some mushrooms that we missed, obviously, at some point, and they're black. Do we do anything? Can
I'll leave it.
spread that?
Oh, well,
So that I
if you
can
want,
more-
yeah, no, if you wanted to, like, it's gonna be gross, but you can, you can take it and just
If I
smear it
get
on a
more
log, and yeah, like it's probably not going to do a lot, like the best way of doing it is making sure you have that hole and then introducing something
Oh, so I have
fresh
to go more
into.
holes and shove them down in there.
Yeah, yeah, like that's probably the best way to do it. Really, so. Really, if it were me, the best way of doing it is to just make new plugs with a shiitake mushroom. So you can actually do what's called a tissue culture sample, and you just take a piece of mushroom, introduce it into plugs, and do it that way. But that way can work. I'm not gonna say it won't work.
And the shiitakes will produce for up to 5 years potentially too.
Potentially, yeah.
Okay.
Again, environmental conditions and specific variety have a lot to do with it, but it is years of production, not a year of production.
So you said your wood dowels that you put in there,
Yes,
is the same wood or
no,
hardwood,
so
softwood?
I use oak dowels, so untreated oak dowels. We use oak dowels just because oak is a really good media for a lot, like a wide variety of mushrooms. And once you have the mushroom inoculated on the dowel, doesn't really matter like what wood is going in the hole, you're putting the mycelium in the hole, right? So that I'm not gonna say it doesn't matter at all, but because the easiest transition you can make from dowel to wood is gonna be the best, right? So if you have oak dowels and you can grow on an oak log, that is going to be the best. But it's not gonna make a noticeable difference if you use a wood that is not of the exact same variety.
Would you say that's the recommended length for your logs or?
You can do it as
bigger,
big as you want.
better,
If you
doesn't
had a
really
tree that fell and you had thousands of plugs, and you
just
really
wondered.
like mushrooms, you could plug the entire tree and it would work. One of the things that we actually like to promote that it's kind of a very specific thing. But if anybody has like a tree stump from, a tree that was unhealthy or whatever, and it happens to be in a shady part of your yard, don't get it ground out, plug it. The root structure of the stump is still alive. It's still vascular. It's still bringing up moisture from the ground. And that just creates this environment that mushrooms absolutely love. So you will get, it goes from a few years of production to like a decade of production.
Wow.
Yeah.
So
Then when you're done, you just have a heap of, mulch. You can just kick over and you don't stump anymore. Right?
So we're constantly seeing different mushrooms popping up.
Mhm.
Like if I showed you a picture like that, would you be able to help?
Maybe, it depends. We can talk after the meeting and I can try, but a lot of times to identify, especially wild mushrooms, if they're a common looking variety, doing things like a spore print, and checking out their gill structure is.
Do they tend to create issues if you're trying to raise your own mushrooms?
No, so what you're seeing in nature, when you see mushrooms grow kind of organically, are billions of attempts, right? Because spores are everywhere, they're all over the place. And you're seeing a few successes, right? So you don't really have to worry about outcompeting too much if you're introducing it in an intentional way. I'm not gonna say it can't happen, but it is, I'm gonna say it's super low risk.
What is the cost to do like a log that size?
This used just under 50 plugs and we sell 50 plugs for, I think it's $12, something like that. Also, but we also do much larger quantities, if you wanted a bunch of reishi for your bees. We produce them by the thousands if you need to. You just tell us the quantity you need, the more you buy, the cheaper it is, because it takes us less time to produce.
I think I found a cheaper hobby.
Jamie, what is the question?
Because I know it has all kinds of anti stuff in it.
Sure.
Does that work? Or-
As far as I know, it should work. I don't. Again, I hate talking in absolutes, especially about things I don't know for certain, but I am not aware of any issues that would help with that.
Don't like walnuts contain some kind of chemical that can cause a lot of things not to grow. Avoid it. Wait,
Yeah, I mean, it's just one of those things that, like I said, I don't know for certain. I've never tried it. I don't know a reason that it wouldn't work. I'm sure that there are varieties that would grow on it, but this is another part that we need to talk about here too. A lot of this goes into understanding your variety of mushrooms as well. So the technique that we kind of described here is only for saprophytic mushrooms that like to grow on wood. There are a lot of mushrooms that like to grow on dung, that like to grow in mulch, that like to grow in, regular soil. So it's good to know your specific variety on what it will grow on. So, yes.
So, the specific question also with mulch thing, are you working with wine cap mushroom?
We do have line cut
You should have
mushrooms,
line cut,
yes, and
okay.
we do, so we're getting the farmer market in May, so. One of the things that we're doing with the farmers market is a different demonstration every week on how you can introduce mycelium, different myceliums and, microcultures into your yard. And that is one of the things that we're going to be doing is how to garden with line cap or the garden giant mushroom.
I've checked into that and was going to do it, and just getting the time to actually do
Oh,
it.
sure.
I didn't get to start that last spring.
Yeah, so
I
I
started
actually,
those last fall, I guess, so I'll be curious to see if they pop up. I'm still waiting for my morels.
oh
They
yeah,
haven't shown
so,
up.
oh man, good luck.
Yeah.
So morels are hard, so I'm not gonna nerd out on it too hard here, but morels love ash and elm trees, and they create a symbiotic relationship with their root structure, and they're content to stay there almost indefinitely until functionally they get evicted, aka the tree dies, right? And then all of a sudden the death enzyme from the tree is what prompts the mushrooms to grow. So seeding your property with morel mycelium can work, but then you also then need to introduce the death enzyme of ash or elm trees. A lot of people do so with actual tree ash. They burn a log of elm or ash tree, spread that throughout. Because a lot of times in nature, you will find morel mushrooms growing after something like forest fire. Anyway, again, I'm not gonna nerd out too hard, but a little bit. Love it. Personally, I think we only have about 15 or 16 right now, but again, we're a brand new company, and if there are varieties that people are looking for, let us know. You know, if you want it, somebody else is gonna want it, and that means that we want to carry it, right?
I
So
think we carry 3 varieties of reishi.
we do. We actually have the regular reishi variety that you saw on the screen. It's just like regular fanned out polypore. And then we have the black variety of that, which looks exactly the same, only it is almost jet black. And then we also have the antler variety of that,
With
which
red reishi too, so we have 4.
by broader.
Nick,
We don't have a storefront right now. We are going to be hitting the farmers market, and we're available online right now. We're also doing different demos at AB Hatchery. We're going to be there in a couple weeks, I think, on the 15th. And then I think Growing Ground, they're Green Top.
Green top on the 22nd.
Green Top on the 22nd. So
We'll
we do
post your
there.
URL here on our Facebook page for everybody, so they can get your.
We also have a Facebook.
Yeah, you can also follow our social. I have a stack of business cards over there. If you guys are interested, I'll hang out after the meeting if you want to talk. And are there any other questions I can address before? I don't know how much time we have. Are we good? Are we good? I'll sit through all day. When they go out in public, like people think I'm nuts because I want to talk about mushrooms so much. You guys want to talk? That's fine. Yeah.
Or should be.
I don't know, this is a good example. So about 3-4 inches.
How many plugs are in that log?
This is, gosh, I want to say it was like 42, 3, something like that. So just under 50, about 50-ish. You can, I wouldn't go much further than this. You could probably space them out a little bit if you really wanted to, but between 2 and 6 inches, it really depends on how fast you want it to colonize and how quick you want it to produce mushrooms.
That little branch
you're a
looks like
patient
a
user.
face.
Would you put it up like that, or you just lay them down?
It depends on how you want to do it. So I'm actually doing some personal landscaping in my yard right now, and I have an area that it's like where the fence kind of meet. My house is at an angle, so I have this area of my yard next to the 6-foot privacy fence that gets 0 sun throughout the entire day. So I'm going to embrace it. And we're putting a whole bunch of logs and totems, which are functionally logs like this, only they're much thicker, much wider diameter, and they're cut into slices. And then you would put your sawdust spawn that we were just talking about in between each layer, like a sandwich. And you can just stand them up like that, and they actually produce quicker because they have that sawdust layer and they don't have to like grow through the bark and colonize the entire log. They produce faster. They start producing faster, but they also expend their energy typically faster. So they will last for closer to 2, 3, maybe 4 years instead of 5+. But so it really depends on how you want to display it. If I was going to do something like this, I would probably bury the bottom third of the log. Because another thing that you need to remember is that even though the log is dead, it's still vascular, which means it's going to draw moisture up from the ground. So if you can get it into the earth a few inches to where the earth is normally at least a little bit moist, it will hydrate your log, which is something mushrooms love. So
Yeah, we've had pretty good luck with the shiitake, but.
yeah, and one other technique
But you're
that,
saying I can't cut my logs now.
what's that?
I missed my opportunity.
Well,
to
it's
cut
not
the
like
logs.
you can't, I just wouldn't, like, it's one of those things, like. So the city, here's, okay, pro tip, guys. The city of Bloomington near Miller Park Zoo has a what you want to call it, like a branch and, like natural waste dump, I guess you call it. And then they turn all that stuff into mulch, and it is accessible to the general public. And that is a great spot to harvest logs. Just go there with your chainsaw, get some logs and go nuts with bugs. But one of the things that we actually, so we do home consultation services for people who are wanting to grow mushrooms in their yard, but are looking for kind of creative ways to introduce them. And yeah, no, I guess that was, yeah, alternatively, yeah, that was kind of my whole point, like, get these and put them in your shade and we'll just produce mushrooms for years.
There's a question online about when you're gonna be at AV Hatchery,
It is the 15th
at what time?
at 9 to 1, 9 to
Tom,
1
is
or
that the
2.
day I think that's the day you do, bees, is it?
Yeah, it's I think the customer appreciation event or whatever it's called.
Well, I was just gonna add something. One of the things that I was thinking about doing with the wine cap mushrooms, we have some large bushes. Yeah, it's terrible. I can't think of the name of the bushes. They cascade over. And so there's this open area in between the front porch, which is the house, and the bushes. I was gonna hide them in there so they stay in the shade.
Yeah,
And,
so-
and then just periodically water them back there.
that's a great idea. One thing to note about the wine cap mushroom too is they're probably one of the more heat tolerant variety and sun tolerant varieties out there. So having them in the shade is a good idea, not always necessary depending on the season. So you will get better production if they are. in the shade.
Watching them online, they talked about, that they'll, if you have them in your backyard, they'll even start growing in your garden.
Oh, yeah.
And that they'll move around.
Oh, so they're called, well, one of the reasons they're called the garden giant is because it's one of the few varieties of mushrooms that really enjoys that diverse microbiome that mulch provides, and mulch is very prominent in gardens, right? And one of the, oh man, guys, buckle up. So one of the things that we haven't really talked about are what's called the mycorrhizal benefits of co-cropping with fungus. And wine caps is a great example. There's actually research through the SARE grant program, some sort of agricultural grant program. I can't remember exactly what it stands for. There it is. They are actually putting metrics behind the benefits of co-cropping with mycorrhizal fungus, specifically the wine cap, and they're showing that they're doing it in kind of a three sister style mounds of controlled and uncontrolled, or controlled and to where the controlled, anyway, the controlled beds are producing the wine cap mushrooms and also showing a between 10 to 15% increase in vegetation yield. So it's one of those things that it benefits the plant, but then the plant also benefits the fungus. So the fungus externally digests their food, right? So they excrete their digestive fluids, and then they kind of suck it up through their cellular membrane, and then they reproduce more hyphae, right? And through that process, that hyphae tries to eat the roots, right? So it kind of pierces the roots on the, you know, microscopic level, and you know, the hyphae are, and they're excreting that digestive fluid into the roots. And those roots love it. It's just high in nitrogen, it's high in the things that they love to eat. So they just suck it up. And they produce additional sugars from the sun through photosynthesis, and they feed it to the mushroom. So it is co-cropping that doesn't have any downsides to either plant or fungus. So that, I think that's just a really cool study. So there you go, wine caps, use them.
Your morel thing is not a support.
No, well, the morel is not a wood loving saprophyte, so it would not grow in a lot like that. Right, so if the morel would want to grow in is on the ground, so you can seed your ground with a nutritious slurry that has morel mycelium in it, and it will increase your chances of getting morel. Right, they're still notoriously difficult to grow in a conventional environment, so yeah.
Five different places.
Yeah.
Okay, Ken, you got a question?
Yeah, you're introducing dead wood, wet.
Yeah.
How do you deal with the ants and the termites?
I know.
Because you're in your yard.
Yeah, I don't really like,
You
so,
just let them live.
Yeah.
To your house.
I mean, no, I don't, so I, not really into my house, like in my house, I use, you know, internal pesticides to keep my ants out. But yeah, no, like in my garden and stuff like that, I love the ants out there. They're really good for pollinating all the other plants that are out
Yeah,
there.
so they start coming to your house, like barrel ants and other ants.
Hey, yeah, that has nothing to do with mushroom lobster. There.
Oh, no, I've just had trees I've cut, the stumps, and then the barrel ants invade the stump, and then they want to come to the house to get the sugars and the grease.
What's the sugar, the grease at the other end of the property?
Well, I think maybe what your question is are they gonna damage your mushroom logs? I
know
can say
that, I'm sure that's not a problem, but it's like when you're introducing things that could damage your house, you can put it next to your wood fence and.
Yeah, I mean, at that point, like, so.
Rural property, knock yourself out, but you know. if you're a town.
You know, I've never heard that as a concern from anybody, so I don't know really how to respond to that, to be honest with you. It's not an issue at my house. I'm not gonna say it's not something that can happen in nature, obviously, and you know, something that could be an ant attractant. But you could say that about a lot of things as well. So.
Well, and what I heard you say wasn't as much just ants as like a pest, like, oh, there's ants there. It was more, I felt like you were more saying the harmful, things, like or ants or termites or things that are going to damage. You know, like, are you inviting and baiting them to be?
Well, you're creating.
Is that what you were
How many
asking?
logs are you generating? You like a raised bed garden, where you've got like 100 logs all laid out, you're watering it.
So how many do I have in my yard, or how many does totally normal fungus produce?
Was typical to use.
So we actually have people, yeah, so we actually have people do mushroom towers, we call them, which are, you know, just mushrooms that are stacked kind of like Lincoln logs, just, you know, 5 or 6 feet tall, and we don't seem to have anybody have issues with it. So.
Yeah, I don't know, I've got a little burn pile of log I burned. Mushrooms are naturally growing.
Oh, sure, yeah.
But every once in a while you end up with a year where they're wet and then.
Oh, yeah.
Ants invade it and they get in everything. You start seeing them in your house.
Yeah.
I mean, I got some good chemicals, but now we're introducing bees. I'm thinking, you know, the great company that makes aspirin that also makes some of the best pesticides. You know, may not be something I want to spray around my house.
Sure, we get those little ants.
Yeah, the little
itty
ants.
bitty ants. Every year, I have found that a mix of like 1 part sugar, 1 part borax, or 2 parts sugar, borax is really good. We put them out, I put out a couple traps here and there.
Yeah,
We'll see them swarm it and then nothing for months. So.
My guess is even without logs, you're probably having.
Yeah.
That's.
Be some chickens in the yard.
Yeah.
It'd be great. I don't know, would allow that, but they don't.
So it doesn't sound like you're too particular about the wax part of it, you're just rubbing a little.
Yeah, I just melted wax over, a burner and, so I have wax daubers, so it's just a piece of cotton on a metal stick, so I don't burn my fingers. And yeah, I just dip it in the wax and, cover the holes, just make sure that nothing that's not supposed to be in there gets in there. And then by the time it starts fruiting mushrooms and the wax naturally deteriorates, it's just not an issue. Because once the log is colonized by a fungus, like it like not always, but it normally will just dominate the entire grow. So you don't really have to worry about it contaminating or getting, something that's not supposed to be in there after it's already established.
Because it's gonna grow from the inside out because of the plug.
Close to the
It's
inside.
gonna infect.
Right,
infect
exactly.
out from
We
all
are
the
intentionally
bugs.
bypassing the bark, which is its first layer of protection, right? So it's like giving somebody an injection of something instead of rubbing it on their skin.
Yeah.
So where do you get your wax from?
PK.
Hey, I found that source first. He doesn't use a hole.
Not yet. No. And
So
that
you
may
start-
change.
So it's only normal, we can't do this in Bloomington or Downs?
Well, yeah, we'll let you.
Okay.
We're gonna go worldwide.
That's the goal. But yeah, it's
So if
been
you-
fun so far.
Yeah, so far you said 22 is when you kind of got going, but like,
Mhm.
how, this isn't something that like just started in 22, like it's got a backstory.
Oh, yeah, so, well, I guess the backstory is 1 year my wife bought me a mushroom grow kit, and here I am. No, really, like, she bought me a mushroom grow kit for Christmas, and it kind of just got me into the hobby, and years later, then COVID hit, and it was at home hobby that I could do that, I could deep dive in, and it just kind of escalated from there.
Could you see the living tree?
What's that?
Could you see the living tree or
You
kill the tree?
could, I don't recommend it because it will likely kill the tree. So, a lot of times when you see, like, I'm gonna use blue oyster for an example. You see blue oyster living on a tree, it's not a parasitic fungus necessarily, but it is saprophytic and opportunistic. So if you're trying to introduce it into a living, healthy, tree that is, naturally resisting, billions of spores on a daily basis, it's probably not going to do much. And if it does, it's only going to eat the parts of it that are likely dead. But once it starts to infect and deteriorate that part, like it can kill the tree. Like it's not healthy for the tree to do to internally inject or introduce a foreign like organism, fungus, whether whatever it is, into the tree, unless it's very intentional. So chaga is a type of medicinal mushroom that grows a lot on birch trees. So there are certain birch farms who will go out of their way to plug their living birch trees because chaga is parasitic and it only grows on things that are trees that are healthy. So it really just depends on the variety of mushroom, your intent, and how carefully you want to deal with it. Which if you're plugging a living tree, I would recommend being very careful. So. Any other questions? Well, it's been fun. Thanks a lot, guys. Appreciate it. Thank you. So I'm not gonna force anybody to take one, but I'll just leave the stack here. So if anybody's interested, feel free to grab one on the way out. I'll clean it later.
Like 9 o'clock, and then we gotta roll because they lock up the door. You can, right at the door, everybody kind of hangs out and talks.
Oh, sure.
Answers questions, so that's a great.
And a lot of visitor.
Okay, it's kind of a so yeah, just, it's, and again, different, a lot of us like those differences and stuff. All right, yeah. Ken, do we want to go with Ken, or do we want to ask questions just for the group, or? Okay, Ken has started a project, he said he'd do.
You volunteered
Attaboy.
me once I
You-
said it'd be a good idea.
Yeah, you came up with the idea, so.
Hey, Ken, give us just couple minutes. Is there anybody that has like a burning question or two real quick? I want to make sure that if you had some questions and wanted to ask, you have a couple minutes and then we'll wrap up with Ken's stuff.
How soon can you split a hive?
That's not a question that can be answered in like a minute.
Okay,
But, there's a lot of things that have to do with it, but next month you'll learn all about it.
and okay, so next month is our splitting top. So our topic next month is how to split a hive. All right,
Well, it's more, it's like swarm management and
okay, swarm management, okay,
If you were to put a queen on it, you could do it as soon as you get a queen, give it a piece.
okay. I got one that's big and busy. I'm kind of like, don't run away yet.
You
Okay,
get the PowerPoint or you need the,
I've
I've got a USB drive where I put it on just. in case
Okay.
you didn't.
There's a question over here.
Oh, just real quick, because I'm getting ready to like get my first bees, like, is there anything that I need to be doing with my hive right now to prep for whenever I put my bees in?
What a great lead in, because that's exactly what he's gonna be talking about.
Sweet.
He's talking about like, what are the April chores?
which one is it?
April items.
Oh,
Yeah, right there. Come by. I got about 1,000.
there we go. All right. Yeah, but this is, our idea is at the end of the day, kind of saying, okay, because it's the end of the month, Ken came up with the idea, we should tell them what they should be doing the next month.
And which
So-
what you should be thinking about.
Yeah, so that, it was a great idea, Ken.
So we'll hit, for you people, your bees are coming soon. Tom, jump in whenever you feel like.
Go ahead, keep talking.
Okay, so your bees are coming soon. Tom, I think April 15th is your delivery date.
That's correct.
A lot of the packages are usually delivered around the middle of the month. So. What do we?
Keep talking.
Anyway, go ahead, just leave that up there so we can,
He's trying to get it so that the onliners can see it too.
oh, okay. Okay, so like I said, package bees tend to come in the middle of the month,
that's like your third slide. Did you want to?
Yeah, because he asked that
Oh,
question.
okay.
I
sorry.
just jumped to that first. As far as installing your packages. If you were in our class, I think we had a part where we talked about installing the packages. If you need a refresher. I'm gonna put the parent document out on this Facebook page, give it to Patty, let her distribute it, that kind of thing. In that document, there's links to both the traditional method, and then there's also a method that Cayman Reynolds put up last year, which was really quite timely for us because that was the first year in 4 years that we needed to get some packages. Basically, what he did was sort of set the package in half of your deep hive, and then sort of let the bees migrate out to the queen from there. Check the video once you get the document there. Don't give your bees too much space. It's one of the things that I've tended to favor at least. If they have too much space, like some people get two deeps and they put both deeps out there. Especially if it's cold. You'll see that the bees might struggle some because they don't have that mass of bees to thermoregulate. So, put your bees out there and start feeding those bees.
So real quick, Ken, I think it's a good comment when you start your bees, you want to restrict the space as well. So like when you're putting your package in, even though you bought a hive that might have like 2 deeps and 2 mediums, all you need is that bottom board, the deep inner cover, and a lid. So you want to keep that space as small as possible. It's kind of like if you were in this room and it's really cold versus being in a little closet and it's cold, you're going to stay warmer in that closet. Same thing. The bees can keep that space a little more managed. And then it also helps them manage the other thing is the entrance, is that entrance is reduced down so they can manage anything that's coming after them.
So a lot of times you'll get, if you buy a beginner's kit from John, he probably gives you an entrance reducer, which is just a stick that long. It's got a small opening, it's got a wide opening, and if your bees are good and strong, you just pull that thing out and let them get at it. Feed your bees with a light sugar syrup. The common thing you hear is 1-to-1, but I've heard a lot of recommendations for 7/10 of a pound sugar to 1 pound water. A lot of beekeepers tend to think that lighter ratio promotes brood growth better. Find a way to get them some pollen. I've seen pollen baskets on my bees already.
I work at Maple's hard,
Yeah,
like it's a cloud around
Is it-
Maple.
Is it light yellow? Like,
I agree.
I'm almost
yeah, real
wondering,
pale yellow. It might be sawdust.
I've got this great big pile of sawdust. I've seen my bees all over that pile of sawdust, so they might just be, I don't know,
My boxwood is pretty light pollen right now.
yeah. But I have noticed some things sort of starting to flower just in the trees.
Yeah, actually, I posted that on our Facebook page. If you look in like the Weather Underground app for your phone, you can scroll down under your local forecast, or even if you're visiting somewhere, you can look in the forecast and scroll down, it'll say pollen. And it's like pollen.com, I think is the website they get it from, but it'll tell you what pollens are active at the time. So that's kind of interesting when you're looking at your bees going, what's yellow or what's gray or what's this. It might give you some hints, and then you can Google what that pollen looks like and maybe get a little better idea.
Cedar, juniper, and elm right now.
Yeah.
Okay, when you get your bees installed, you'll be feeding them. But give them about a week to sort of get used to their new home. You get them too quick and they might decide, hey, we get no privacy around here, and they'll just abscond. So give your bees about a week at least to adjust. Do you agree with that, Tom?
Yeah, I would say, like, to put it into an analogy, like if I threw you in an apartment with no furniture, no pots and pans, no anything, and said thrive. Versus if I put you in an apartment with some furniture or food in the fridge and stuff like that, you're probably gonna be more successful in that second situation. So as new beekeepers, you know, a couple of you are just getting started, you go buy a kit, you know, all your hive gear, you're not gonna have drawn comb. So you're at a deficit there. So the ticket there is just make sure they got feed, and they can build up some of that wax themselves and manage that space. So definitely, please feed your bees. I hear a lot of people are like, oh, I want to be natural, I don't want to get many additives, I don't want to get many feed, I don't want to do any of that. If you have a baby, you feed your baby. If you get a puppy, you feed your puppy. If you get a cat, you feed your cat. It's the same thing. Make sure they have access to feed during that time.
And that's with colonies too, right?
Yeah, I would say new and old, actually.
Well,
even
new
in the
because
spring.
then like getting the nuke as opposed to like the package.
Yeah, so the tricky bit, and you're gonna learn more about this in our, I think it's our May meeting, is we're gonna go over like inspections and how to do that. Plus, so the thing I didn't say in the beginning that I always say is right now, the reason why we're here and the reason why we have bees and brews, even whether you drink or not, come to those things and shake hands, meet some people, shake hands, trade telephone numbers, and make some bee buddies. And even if you're both brand new, you can make a pact and say, hey, I'll come over and play bees with you, and you come over and play bees with me. And it makes you accountable. So that's yeah, there you go, bee buddies right there. That's really important. You're gonna learn something from each person you go play bees with. And you'll get exposed to different equipment and different tools and different techniques and stuff like that. And make as many contacts around here and go work bees with everybody you get a chance for. Now that said, if PK is doing regular inspections on the club hives, or you know, myself or anybody else here who has bees that overwinter. Okay, there you go. Those are the people who go tap them on the shoulder and say, hey, can I come help you put a beer together? Can I come do some work? Can I help? You're gonna sweat and be a little miserable with it, but you're gonna learn, right? You're gonna learn during that time. Learn on their dime. So come work these with them, because they need a hand as well. So, sorry, I went on a tangent here, but
Yeah.
make friends and go work together. But
Okay,
you'll see this stuff in action.
okay, I'm trying to rush it some to keep us on track here for time. Most likely won't happen in the 2 weeks that you have your package, but be looking at them to see once they get about 6 frames drawn out and some work being done on it, that's when you can consider using the second D. I'll jump back to the first year overwintering.
Wait,
people.
and just clarification, 6 frames means the front and back, like both sides of the frame.
Yeah, that's. We were being more cautious the very first time we got these. And we went to about 8 frames. And come June 1st, I saw a whole bunch of bees swarm. So. you don't-
So on that, I'd say more like 60%.
Yeah.
Because some of us run 8-frame mediums, not 10-frame deeps.
Yeah, especially if you have just foundation out there. They're gonna wanna have, because if they get to the point where it's all drawn out and they're filling it up with nectar, even if you've already just put another deep full of foundation,
And we're gonna.
they're
deep
not
dive
gonna
into
have
this
that.
next meeting.
Yeah, okay. Just quickly for the people that have been in last year's class and first year overwintering, you should be feeding them light sugar syrup right now. We've been doing it for about a week. We could have been doing it a little bit longer. Eggs laid April 1st will be emerging from the cells around April 21st. And it's another 15 to 25 days till the bees can fly and collect nectar. You want to build up as much population as you can for nectar collection as you can. Inspect your hives on warm sunny days. Look for potential problems now. I've seen a few frames or a few hives that are on the weak side. I've got a few hives that are on really strong side. I've already moved 2-3 frames of brood into 3 weaker hives. Wait to rotate your hive bodies. Probably closer to the end of the month or the start of May.
When your nighttime temps hit 50, 60 degrees consistently.
Yeah. Don't be in a rush to remove whatever thermal mitigation. In other words, if you got insulation on those bees, don't be in a rush to take it off. And if you are getting antsy and wanting to do it because it's just a pain to take it off, go through your hive and then put it back on. At least look at a 10-day or 2-week forecast. Get your swarm traps out. Because if you're feeding and Sharon's really worried that I'm feeding our bees too early, she's afraid they're gonna swarm on us.
So do you feed both your bees or just yours?
I don't touch her. Unless she
What I told you to do.
Unless he says, I'm too lazy to go out and do it and it's cold outside, can you feed them for me? Okay, hive inspections, and this is once again mostly for the people that are first year beaks. Have your smoker ready. It helps calm them down a little bit. Especially if it's cold or feisty. I have what I call a quiet box. And in my case, I use a Pro Nuke. And it's a nice light little piece of plastic hive that I carry around. And if I start my inspection, I pull the first frame out. I put that in the Pro Nuke. And that way I've got room to work in my hive.
Why do you do that? Like, what's the room for? What's your reason? Tell them that.
Well, you've heard people talk about rolling a queen.
What does that mean?
Rolling a queen means that you're sort of clumsy, you sort of pulled the frame out too fast and the queen was there and she just got rolled up in the whole situation and she gets squished. You don't want to squish your queen at this stage of the game. You don't ever really want to roll your queen. But the other thing is I also keep one of these little, they call them a hair roller type queen cage. They're just a little round plastic queen cage. And there's times I might say, I found the queen, first frame I go into, I'll put her in the queen cage, put her in my pocket. And now I can be a little clumsier. So, you know, but
It's a great idea when doing your mic testing.
yeah, especially when you're doing mic testing. If you find that queen, she either goes in the quiet box on her frame or she goes in the queen cage. Now, this is a thing that I'm guilty of doing many times. I'll go out there with my bee suit on. I don't flip my hood up until I get close to the bees. I flip up my hood and then I forget to zip it. That's not always fun if they're feisty. Look where your bees are clustering. I look at my, you know, and I take that top off, you know, I'll give them a little puff of smoke, sort of get them come or let them know that I'm coming in. But then I'll take that top off and I'll look and say, I see most of the bees over here. This will be the frame that I pull off first. The reason being is most likely that heavy area of bees on the top of the frames is where most of the brood is. You start here. Once again, you're minimizing your chances of rolling a queen. Now that isn't always a sure thing because I found queens on that spot where I didn't expect to find her, but at least improve your chances. I leave the first frame out while completing my inspection. I talked about that. Now, you want to look for evidence of a queen. Do you see eggs? Do you see larvae? Do you see capped brood? Do you have drawn comb? That's where it's all got the little honeycomb things there. Is there open space on that drawn comb? Because if you don't have open space, you'll be having to worry about swarming problems. But that's the things that I'm looking for. Do I need to do something to give them another box? So be ready for plan B. And that goes into your swarm control. Tom will be talking about that more next month. Is that right?
Yep.
And take notes. Lots of different beekeepers talk about you know, writing on the top of their hive. It's not a bad idea to take a Sharpie with you and write on the top of your hive. I need a frame of brood on this hive. And by the same token, you can say on this drawn hive, plenty of brood here on this hive, come to me to get more brood. So sort of rush through what I have. This document that I'm sending is like 4 or 5 pages long. So it's sort of like the fire hose that you guys have complained about when you took your class. But this is sort of go time. If you're getting a nuke, you may not be getting your nuke until the start of next month. But
For me, it's at the moment.
you'll be getting a leg up in some ways. It's a little bit of an insurance policy, just in the thought that they've already got a running start. They're less likely to say, oh, I'm in an empty apartment with just a mattress to lay on. So but the nukes the packages will help you get things going.
So I got time for like 1 or 2 questions based on some of the stuff that.
Was there anything else in the chats that anybody, okay.
Makes sense.
I'll send this out, both email, we'll put it on the website, we'll put it on Facebook, so you can have a chance to digest it too.
Yeah, Patty, Sharon noticed some typos when she read my document, so give me a chance to,
Too bad. Okay.
I'll make some corrections and I'll send you a new PDF.
He gave it to me. What can I say?
Yeah.
Well, it may be stupid, but I really do
You're too bad.
let her go over my stuff and she says either she's my worst critic or my best critic. I never quite figured it was.
Yeah, so actually, this is a really good question that he's asking me that probably a lot of you might want to know. So I'm gonna call you out here. So he's asking me, he said, I had some colonies die. They got a bunch of honey. What do I do? And he said, do I leave that honey to feed the bees? Or do I take that honey and enjoy it, or somewhere in between? And my personal opinion is, I said, extract that honey and enjoy it. Some others may disagree with that. Here's my reading. I'll tell you that. And I'm not saying mine is the only way. But for me, what I would say is extract that honey. And if you don't want to eat that honey, you can dilute that honey a little bit and use that as feed. But what I would say is I rarely have my bees eat honey. It sounds weird. Like, I'll have a cap frame of honey and I'll put it in with my bees and they won't touch it. Now, my Amish friend taught me a trick where if I take my capping scratcher and I scrape all that wax capping, then they'll go after it sometimes. But what I would do instead is I would thin out that honey, or I would I'd feed sugar syrup, because I know that doesn't have contaminants, or doesn't have nosema, or doesn't have any of those other things.
Right.
So I would tend to feed sugar syrup because I know it's clean. They're gonna be getting pollen from stuff around, or my pollen patties or things like that to fill their protein needs. So that's what I would do personally. Some others may disagree.
Yeah.
That's my thoughts.
My preference is that right now I want them thinking there's a flow going on from that light sugar syrup.
So what Ken's trying to do is he's trying to stimulate them to build. And that's, when I say build, that means draw wax, lay egg, and then still have feed to feed the pupa and larva. Sorry, the larva.
I want my flyers ready to go when the actual nectar is there.
Yeah, so what Ken's doing also is he's dialing back from his big flow. So he knows when his big nectar flows are, because he's been doing this for a while. And they pay attention to this. Because that's where they raise their honey, and that's the business. So they pay attention to when their flows are in their area and their neighborhood that their bees work. And then they dial back 6 weeks. You say, why 6 weeks? Well, it's 3 weeks for the bee, from the time the egg's laid until the bee comes out. And then it takes a couple of weeks to mature and be a forager. So there was somebody else that asked a question. I think it was on the Facebook forum or something like that. They're asking about how long until drones, because they're like, well, if I raise my own queen, is it a good time right now? And somebody said, make sure your drones are sexually mature. You're like, I don't know what that means. So that's a different story. But the time period is from the egg being laid till they're sexually mature is in 5 weeks. So if you're planning on doing a walkaway split and you don't have drone comb in your hive, now is not the time to do that. And we'll deep dive into that next month.
It's interesting. I'm seeing some drones walking around the hive already.
I have two.
So,
Okay, there's a question over there.
Yeah, it's too late to order.
That's your man.
Yeah, I'll give you my number. If I can't get you, I'll get you to somebody who can.
Good.
And we gotta wrap up. So we get the hook.
Employed.
I have one like 2 weeks ago when we were checking. Check
We're allowed to plan or What did you think of the mushroom presentation?
Right.
Irish or?
So, thank you.
Okay, they're good.
Right.
I know you sound like.